A new study says your cells are ticking time bombs, and somatic mutations alone could cap human lifespan at 146 to 194 years. Researchers have done something never attempted before. They have quantitatively assessed how somatic mutations alone limit how long humans can live. The answer is not indefinite. The answer is roughly 146 to 194 years. That is about twice what we see today. But it is still a hard ceiling. The study was published in NPJ Aging by a team at the Skolkovo Institute of Science and Technology and the Artificial Intelligence Research Institute in Moscow, Russia. Led by Evgeny Efimov and colleagues, the paper is titled Somatic Mutations Impose an Entropic Upper Bound on Human Lifespan. They built an incremental modeling framework that progressively layers in factors of aging to see how population survival dynamics change, and the results reveal a striking asymmetry across organs. To understand the impact of somatic mutations, the team first asked a counterfactual question. What would happen if we eliminated every single hallmark of aging except mutations? In that hypothetical world without any aging at all, the theoretical baseline for human lifespan was set at roughly 1,759 years. That number comes from their survival dynamics model. But when they added back only somatic mutations, the median lifespan collapsed to 156 years. That is a drop of over 1,600 years from a single factor. It shows just how powerful mutation accumulation is as a biological timer. The study found a fundamental asymmetry between cell types. Post-mitotic cells which do not divide are the critical longevity bottlenecks. Neurons in the brain and cardiomyocytes in the heart fall into this category. Because they never get replaced, every somatic mutation that occurs in them persists for the life of the individual. Over decades, these mutations accumulate and degrade function. The brain and heart cells are the main bottleneck for longevity. The liver, by contrast, is made of proliferating tissues that constantly regenerate. Cellular replacement effectively neutralizes mutation-driven decline, meaning the liver could theoretically last for thousands of years, even as mutations pile up. The methodology behind these findings is what makes this study stand out. The researchers developed an incremental modeling framework. Instead of looking at aging in isolation, they progressively incorporated factors into a model of population survival dynamics. Each aging hallmark was layered in one at a time. This allowed them to measure the independent contribution of somatic mutations to lifespan limitation. By comparing models with different combinations of aging factors, they could isolate exactly how much mutations alone reduce longevity. This step-by-step approach gives a level of quantitative clarity that previous studies lacked. When the team ran their multi-organ integration model, the predicted median lifespan landed between 146 and 194 years. That is approximately twice the current human longevity. But here is the crucial insight. The study's authors emphasize that this substantial yet incomplete reduction indicates that somatic mutations significantly drive aging, but cannot alone account for observed mortality. In other words, other aging hallmarks are contributing comparably to the aging process. Fixing mutations alone would roughly double your lifespan, but it would not make you immortal. Other factors are just as important. The Reddit discussion on the science subreddit has some notable reactions. One commenter noted that the first step to solving a problem is identifying it, and this study does exactly that. Another joked about turning all our cells into liver cells, since those could theoretically last for thousands of years. Someone else pointed out that even if we figure out how to grow organs using our own cells or build artificial hearts, the cardiovascular system as a whole presents challenges, with veins and arteries thinning and filling with plaque. One commenter raised a practical concern about inequality, worrying that longevity extensions might only reach the richest people for much longer. Another noted that if we do extend lifespans significantly, cancer becomes the #1 cause of death, since everyone would get cancer over that extended course of time. So what does this mean for the future of human longevity? The takeaway is clear but nuanced. Somatic mutations are a fundamental biological timer, and they alone impose an entropic upper bound on how long we can live. But they are not the whole story. The brain and heart are the critical bottlenecks because their post-mitotic cells cannot be replaced. The liver and other proliferating tissues could last for thousands of years through cellular turnover. To push beyond the 194-year ceiling, we would need to address multiple aging hallmarks simultaneously. This study gives us the first quantitative map of where the real limits lie. The full paper is available open access through NPJ Aging. Thank you for watching. If you want more deep dives into longevity research, subscribe and hit the bell. If you enjoyed this deep dive into the science of aging, subscribe to Orbit Reports and hit the bell. We break down the latest research every week. See you in the next one.